The aim of this procedure is to use the fluorescence in situ hybridization technique to determine the sex chromosome or translocation status of in vitro human embryos at risk of genetic abnormality. This is accomplished by lysing a single cell biopsied from a day 3 embryo onto a glass slide and air drying the nucleus. The second step is to co-denature and hybridize the target nucleus DNA with DNA probes labeled with fluorochromes of different colors. Next, excess probe is removed using a stringent wash and the nucleus is stained with a DNA-specific fluorescent stain. The final step is to view the nucleus using a fluorescence microscope and to capture digital images. Ultimately, results can be obtained that show the copy number of the targeted chromosome regions through fluorescence microscopy.
Generally, individuals new to this technique will struggle because it is at the very limit of the fluorescence in situ hybridization technique.
"24 hours in advance, prepare the cell lysis buffer for spreading cells. Filter the solution and dispense 1 ml aliquots into 10 to 15 1.7 ml sterile microcentrifuge tubes. Close and label the tubes prior to freezing at -20 degrees Celsius. Just before the biopsy procedure, defrost the lysis buffer at room temperature. Score a small circle, approximately 5 mm in diameter, on the underside of an amine coated slide using a diamond pen, and then, using a hard 4H pencil, pre-label the slide. Blot with a latex glove to remove any graphite dust. Use a separate slide for each blastomere in numerical order. Now, place a small volume of lysis buffer within the circle. Transfer the biopsied blastomere into the lysis buffer. Add lysis buffer as necessary to lyse the cell. Observe the nucleus to ensure that it remains within the circle and is not lost. If the cell does not have a nucleus or has multiple nuclei, biopsy another cell. Leave the slide to air dry at room temperature. First, pre-wash the samples in Coplin jars using phosphate-buffered saline for 5 minutes at room temperature. Then, rinse twice in sterile distilled water. Dehydrate with an ethanol series for 2 minutes each at room temperature and air dry. Ensure slides are fully immersed, and if any graphite dust floats to the surface, soak up with a clean tissue. Record the position of the nucleus within the circle by visualizing with a phase contrast microscope. Then, dehydrate the samples with 100% ethanol for 2 minutes at room temperature and air dry. Now, apply 2 microliters of probe mixture and cover with a 9 by 9 mm cover slip. Seal the edges of the cover slip with rubber cement. Perform the co-denaturing step on a heat block at 75 degrees Celsius for 5 minutes and then hybridize overnight in a humidified chamber at 37 degrees Celsius. For each Coplin jar, equilibrate 50 ml of 0.4 times SSC stringent wash solution in a 71 degrees Celsius water bath. Place the stringent wash bottle and the empty Coplin jar in the water bath at room temperature, heat to 71 degrees Celsius, pH the stringent wash, and decant into Coplin jars. Proceed to carefully remove the rubber cement from each slide and rinse off the cover slip using 4 times SSC 0.05% tween 20 pH 7 at room temperature. Wash the samples in the 0.4 times SSC stringent wash at 71 degrees Celsius for 5 minutes, followed by 4 times SSC 0.05% tween 20 at room temperature for 2 minutes. For probes that are indirectly labelled, drain the slides of excess liquid and apply 20 microliters of fluorescently conjugated antibody under a 20 by 20 mm square of parafilm. Incubate in a humidified chamber at 37 degrees Celsius for 15 minutes. Remove the parafilm and perform the following washes at room temperature for 2 minutes. once in 4 times SSC 0.05% tween 20 and twice for 2 minutes in PBS. After draining the slides, apply 6 microliters of Vector Shield mounting medium containing DAPI to a 22 by 22 mm number 0 cover slip and invert the slide over the cover slip. Blot and seal the edges of the cover slip with clear nail varnish. Finally, analyze the samples by fluorescence microscopy. Score each nucleus by 2 independent analysts. Also, use imaging software to capture an image of the nucleus for confirmation of the visual diagnosis, and for image archiving as part of laboratory quality assurance. Here, a metaphase spread and an interphase nucleus prepared from cultured peripheral blood lymphocytes indicate a carrier of a reciprocal translocation between the short arms of chromosomes 5 and 9, with breakpoints at 5p14.3 and 9p24.1. FISH probes illuminate both translocated segments and the centric segment of chromosome 9. Signals in interphase blastomere nuclei from day 3 embryos can be captured and then merged to form a composite image. 2 signals for each probe indicate 2 copies of each locus, which is consistent with a normal or balanced complement for the translocation chromosomes. Coloured signals indicate 1 copy of the translocated segment of chromosome 5, 3 copies of the translocated segment of chromosome 9, and 2 copies of the centric segment of chromosome 9. This data is consistent with an unbalanced product of monosomy for 5p14.3 to 5p ter and trisomy for 9p24.1 to 9p ter.
After watching this video, you should have a good understanding of how to lyse a single cell biopsied from a human embryo and prepare a single nucleus using the fluorescence in situ hybridization technique. This is important to achieve reproducible and optimum test results to determine sex chromosome complement or translocation status of an embryo.